Okay, today's lecture is entitled Evolution of Populations. So we've already talked about Darwin's theory of natural selection, and now we're going to see the mechanisms that are going to be used that actually promote this change over time. Okay. So just to get you back into the ecology mindset, we're talking about populations here. So this is a group of individuals. Okay, a single individual does not evolve. Rather, a group or a population evolves. Populations are individuals that come from a same species. And remember, what defines a species is that they interbreed and produce fertile offspring. Remember, we did that activity looking at the isolating mechanisms, which determines when two species are still two separate species. In this case, in order for us to look at the change over time of a population, we have to be looking at the breeding within a single species. Okay, so within a population, members are interbreeding, and so therefore they're constantly swapping genes back and forth. Okay, we call that a gene pool, and the diagram up here is showing you here are a bunch of organisms, and these are their genotypes. Some of them are, homozygous dominant, homozygous recessive, or heterozygous. And then through random mating that occurs in the population, we're going to mix up the possibility of bringing these different alleles together. So all of the alleles within a population are called a gene pool. And I really like this diagram because it helps to pull the vocabulary term and match it with, what it means, okay? The gene pool, right? Right, so the gene pool consists of all the genes present in a population, including the different alleles. So this will be both the big B's and the little b's, all alleles from any genotypes. Obviously, if we're talking about different types of traits, for example, if some of them are polygenic or if there are multiple alleles, as many alleles as are in the genotypes will go into the gene pool. But we're just going to stick to a basic Mendelian monohybrid example where there's one dominant allele and one recessive allele to keep things kind of simple for this explanation. Okay? So when we look at all these alleles, we can count up how many there are, okay, and we can measure them relative to the total number of alleles within that population. And remember, this is at one gene. So the allele frequency is the number of times an allele occurs in a gene pool. So here, for example, okay, we have a sample population. When we look at the black mice, okay, the number of individuals, 48% of them are heterozygous black, 36% are homozygous brown, and 16% are homozygous black. Okay, so there are three different genotypes, two phenotypes, and there are two alleles that we have to look at. So when we start adding up the amount of alleles, we need to take two alleles for each individual from the homozygous black, and then also one allele for each individual from the heterozygote because they're going to carry one of those alleles as well. Okay. So what you could do is you could count up the number of alleles and then we express that as a percentage. So for example, in this population, 40% are possessing the dominant big B allele in some way, and there is a 60% frequency of the little b allele. Okay, remember the little b allele appears twice in homozygous recessive individuals and once in heterozygous individuals, and the dominant b allele appears twice in homozygous dominant individuals and once in the heterozygous individuals. Okay, so all we're doing is we're counting how many alleles there are of each type within one gene of a sample population. And we're going to look at how that changes over time. The frequency of the allele has nothing to do with whether it is dominant or recessive. Okay, for example, in this population, it just so happens that the recessive allele is more frequent. We talked about this a little bit in chapter 11 when we talked about polydactyly, which is a recessive condition, or I'm sorry, a dominant condition in which you have multiple fingers or toes above the 5 digits that you have, so 6 or 7 is the most common form of polydactyly. That is a dominant trait. However, how many people do you know that have polydactyly? Not that many. So what that's an indicator of is the fact that in our population, our human population here, we have very few dominant alleles and very many more recessive alleles. So when we would look at the frequency there, the recessive allele frequency would be much higher. Okay. As I just said before this, when we change that frequency, okay, when the percentages start adjusting, either up or down or evening out in some way, it doesn't matter, then that is a form of evolution. That is how we're going to change. So, for example, we were at a 60-40 split before with our alleles here. 40% were black and 60% were brown. But if we saw the black allele start dropping, okay, and if its frequency went down to 30%, then that's evidence that there is some evolution going on within the population. Okay. Now, when Darwin was coming up with all of this idea, he had no understanding of genetics or DNA. He certainly did not understand anything, relative to mutations as specific within the DNA code. But he did know that there was variation, and he knew these differences occurred and that they were pressured by the environment, changes in the environment, random mutations occurring. We now know those random mutations occurring have to be having an effect on your nucleotide sequence in your DNA molecule. Ways that we can get mutations, we've already gone over this, but just a quick review. There can be some mistakes that occur during replication, okay, so those are what we would consider spontaneous errors. Or we know that we can induce mutations, for example, through the use of chemicals. That was the seedless watermelon example, okay, and also through the use of radiation, and that was how we were able to mutate the oil-eating bacteria. So we can force these changes, or they can happen naturally, okay? But the changes that occur within the nucleotide sequence of our DNA then affect our genes, which affects the number of alleles that are going to be relative in each individual within a population, and that is going to produce variation. How does it produce well, it produces the variation, and then nature acts on that variation. So we've talked about Darwin's theory and how survival and reproduction are going to be affected by the different variations you have. For example, some rabbits are able to run faster than others. That ability to run faster comes from a genetic change that was the result of a random or an induced mutation that caused their gene sequence to differ, which produced a new phenotype. All right, so here we have a little comic strip. I don't know if you can see in the picture down here, the farmer saying, "Who's it gonna be? Who's it gonna be?" And you'll notice that all of the chickens pretty much look the same except Mr. Longneck here, okay? And if you know anything about harvesting chickens, they have to cut their heads off. So if he's randomly selecting a chicken, okay, that chicken's phenotype is going to have an adverse effect on his ability to survive and reproduce, because it's, in a joking manner, making him more likely to be selected for execution. Okay. A lot of times students get this as a misconception. So big star on this slide, little note to yourself here. When we talk about variation, yes, crossing over, sexual reproduction, the formation of your gametes, independent assortment, those are all a source of variation for the population, okay? It produces a variation, but it does not affect your allele frequency. And I love this analogy. I think it puts it perfectly, okay? You have a deck of cards, and the deck of cards has a certain amount of numbers and a certain amount of suits, okay? As you shuffle the deck and randomly pull two cards, you are always bringing together, a different combination. However, you're not changing the amount of, say kings or queens that are in the deck. So the number of alleles stays the same. The random combination of those alleles coming together through sexual reproduction, that's going to produce variation. Very important for you to understand, allele frequency is not changed by sexual reproduction. Therefore, evolution is not going to occur due to increasing sexual reproduction. Okay. All right, so now that we have covered the basics of allele frequency, let's look at some things that can cause our allele frequencies to change. Okay. Natural selection is obviously going to be a large part about changing allele frequencies, and this will lead to evolution of a species. Here's a great example, okay? Let's say there's an initial population of lizards and they are mostly brown. But through random mutations, okay, we have introduced two other phenotypes. Therefore, there are new alleles that are present through mutation, and that has affected the allele frequency. Okay, this is our initial or our starting population. So we have 80% brown, 10% red, and 10% black. Well, now let's put them in a situation in their environment, and hey, red lizards, they would stand out in a forest ecosystem, so that would mean that they have a higher chance of being prey. Therefore, they are less likely to survive and reproduce based on, action in their environment. Okay? And let's say that the black lizards are able to absorb heat faster. So as we know, lizards are cold-blooded. They need to take time. They need to regulate their body temperature. Black lizards can do it faster. Therefore, they're able to process their food and avoid predators more often than any of the other colors. So their survival is going to increase. If we see an increase in their survival due to that, we're going to see an increase in their alleles. Just like when we see a decrease in the survival of the red lizards, we'll see a decrease in their alleles. So we can look at how over generational time this is going to cause populations to evolve. Okay. In addition to natural selection, we know that sometimes when you put pressures on the probability of genetics, it's going to affect evolutionary change as well. So for example, if you have a very small population, then the predictions you could make about how that population will respond to change won't be as accurate. Think about the same thing. If I flip a coin 3 times, well actually let's do an even number. If I flip a coin 4 times, okay, it should be 2 times heads and 2 times tails. Much less likely to actually get a 50-50 split. However, if I flip a coin 400 times, I have a higher probability of getting much closer to that 50-50 split range. So now, being a very small population is going to affect how we can predict things. Okay? Genetic drift in small populations is the random movement of alleles in or out of populations, and this can happen when we colonize a new habitat. Okay, so again, genetic drift is just random changes that happen in the frequency of the alleles simply because the population is so small, okay, that we're not seeing the predicted outcomes. So here's an example. Let's say we have an original population here, okay, and two groups decide to go off and journey on their own. So they're going to start their own population, or they're going to found a population. It just so happened of this entire population, the two that start population A here are of the dark and the yellow dotted variety. And the ones that start population B are of the red dotted and the yellow dotted variety. So now when we look at descendants and we go through generation after generation, we're going to have a change in the allele frequency compared to the original population. Okay, so these individuals were sort of randomly selected, okay, but because they were so small, they weren't a representative from each of the different types that were in the original population, and therefore their allele frequencies will be different. Same thing for population B. And again, this affects small populations that are migrating to new habitats. We call this a founder effect. Okay, so a founder effect is the result of genetic drift, a change in allele frequency due to a very small population. This is one of the things that can cause evolution that has nothing to do with natural selection. Okay, there's no pressure in the environment here to be brown or yellow. It just so happens that, the founding population only were brown and yellow. Okay, that would be just like, humans colonizing a new land. Okay, I'm sure that, our when our colonists came over from England and from Spain, they were a small, select few population. Okay, they were not an equal representative of everybody from England and Spain and France and things like that. And so because of that, that founding population, that dictates the allele frequencies of the coming generations. Okay? So what we're going to do in class is we're going to do an activity so you can see how allele frequencies can change over time. They can change, again, due to natural selection, as well as due to, variations in populations that are small. For example, genetic drift that results in the founder effect. All right? If you have any questions, please email me or see me before or after class.